It receives from the stomach chyme. That's what we call that churned material. It's acidic. So this is chyme coming from the stomach. It receives from the liver via the common bile duct. The most important component is bile salt. There are additional waste products, bilirubin from breaking down the red blood cells and so on. But as far as digestion is concerned, the primary component are the bile salts, which the stomach has broken down the lipids into small lipid droplets, which would recoalesce and form larger lipid droplets, but they become coated by these bile salts, and that repels them from each other and maintains that emulsification. And then joining the bile salts coming from pancreas, we have the bicarbonate ions we talked about that you lose with diarrhea, and we have our digestive enzymes, which are more important than the stomach and mouth digestive enzymes. So bicarbonate ions and digestive enzymes coming from the pancreas. So this duodenum is so short, and you may say duodenum, I just say duodenum. It's so short that there really isn't insufficient time for digestion to start here. This is just where this gets mixed up and where the chyme is neutralized to an alkaline pH that is required by the pancreatic and intestinal enzymes. Then this duodenum becomes intraperitoneal. Well, it doesn't become intraperitoneal, but it gives rise to the jejunum, which makes this right angle turn as it comes into the peritoneum. And this now is our jejunum. Between 8 and 9 feet long. And this is where we do a lot of the secondary digestion. So when we talk on Thursday next week about breaking down food, we take, for instance, if we're talking about starches, remember I said if you chew a cracker long enough in your mouth, it's going to start tasting sweet. That's going to take place instead with salivary amylase, it's going to take place with pancreatic amylase. It's resulting in disaccharides, which are too large to cross the plasma membrane of the cell. But in the jejunum, it's primarily where we have the digestion occurring with the pancreatic enzymes. And then towards the end of the jejunum, those products, the disaccharides or the short chains of amino acids, will interact with enzymes actually made by the small intestine. And we have single sugars and single amino acids that can be then directly absorbed into the body. And that's what takes place at the ileum.
Is this retroperitoneal or is this non-peritoneal?
Both of these would be intraperitoneal. Thank you for asking. Okay, so there's an overall view of the 3 regions. And now let's go back to the GPAT. Again, this is what we looked at yesterday. So we're not going to make any modifications. All right, I'll emphasize some specialties by region, but we're not making any modifications because that was our general plan in the first course. May I erase what's on the board?
I'm slow because I'm like doing the camera. change you.
Okay?
Just ilium, hold on.
Ilium and entropy,
Okay, got it.
not entropy, but. So our mucosal plan, or our mucosal organization, we have villi, and we have intestinal glands, like you're over there on the board. The epithelium is simple columnar. And now we have goblet cells. So this is the mucosa of the small intestine. On the GPAC, so our epithelium is still simple columnar. But now we see goblet cells. We didn't see them in the stomach. So this is the source of our mucus secretion.
So are we doing it by section like this?
And we mucosa, submucosa.
No, I'm talking about duodenum.
No, I'm gonna talk about the specializations or uniqueness within that.
Okay.
Okay, for each of those, if that makes sense. So here you can see the goblet cells. The goblet cell, well, I'm not sure what you mean by that. The goblet, yeah. But we see a lot of goblet cells. The goblet cells in the small intestine and the mucus secreting cells of the stomach are protective against the digestive nature of the folic acid and the enzyme. When we get to the large intestine and rectum, we see even more goblet cells, not because of more digestion,
More secretion.
but for the lubrication. Okay? So let's put some goblet cells in here. So this structure, again, that I just drew, is identified as a villus, not to be confused with microvilli. It contains within it loose, irregular connective tissue. Remember what the term we use for that? Again, lamina propria. And we have a unique lymphatic capillary that we associate with villus, the lacteal. And the reason that this is so important in the digestive system is that our single amino acids and single sugars are going to be picked up into the capillary system, but our lipids become coated with the protein. That's what makes them soluble in the water of blood. Otherwise, a lipid would not be soluble.